On behalf of the Genome Sciences Alumni Board, thank you all for coming. We're pleased to welcome back Genome Sciences alum, Dr. Ivan Liachko, for this week's Wednesday evenings at the Genome. Dr. Liachko received his PhD from Cornell and then was a postdoctoral fellow here while co-founding Phase Genomics, where he is currently the CEO. We'll be hearing about the exciting applications of the technology being developed there for illuminating the dark matter of the microbiome.
Cool. Yay. Thanks, everybody. Thank you for coming. I can see that almost everyone here, I know. So maybe I need to switch PowerPoints real quick. I thought there was going to be like a lay audience. Thank you for introducing me. Thank you for having me in this lovely air-conditioned building. I was going to give a talk. I titled my talk about microbiome stuff. I'm still going to talk about microbiome stuff, but then as I was sort of putting it together and I was thinking about kind of how to theme it, really, I kind of pivoted a little bit, and so it's really going to be about crypto and supplements and how to sell them. No, no. So my theme today is actually going to be information. Now, so most of you have seen my talks because you've seen them from before, so you know sort of roughly what I'm going to talk about. But as we've been in doing what we do for a long period of time, I get to like wax a little bit philosophical, and there's now enough sort of different things that we've been working on over the years that kind of tells a story in a slightly different way. So for those of you who don't know me, I was a postdoc here, and about 10 years ago, which was, we were just discussing with Maitreya. So I was in Maitreya's lab, and I worked with Bonnie and Raghu, and I worked with other folks in the department. We collaborated with Jay Shanduri a lot. It's relevant for this talk. And so 10 years ago, we had this interesting idea, and we started working on it. And then it bloomed into this company that is still around 10 years later. And the company has kind of changed around what it does. And also a little bit of the story is going to be about kind of like a little bit of history behind it, but it all has sort of a unifying theme. And the theme is information. So you will notice, because I'm a CEO of a startup, or it's not a startup anymore, but it's a small company. I make slides like this. I talk about how life is actually information. And it's actually true. It's not entirely just woo-woo. So, and of course, the genome is the blueprint of life. And then I was those of you who don't know, right? So DNA coils up, turns into chromosomes, and this is what encodes most of your biological functions. And because I was coming out of this department, and because at the time, this was still a long time ago, you were still allowed to show this figure. You can see the figure stops in 2022 because that's when it became illegal to show this. But the idea is, at the time, sequencing was just like everybody wants to sequence everything. You want to sequence genomes, and when you're sequencing something, when you're trying to put a genome together, you have to go through this cavalcade of assembly, right? So you have to. So for the hypothetical layman audience that would have been here, you have to. I know I see some folks, but right. So for those of you who don't know, right, a genome is this big thing with chromosomes in it, right? You want this thing, but when you sequence, the sequencers don't give you a chromosome. They give you these little short bits called sequence reads, which you then assemble into slightly larger pieces called contigs. But eventually what you want as much information from the genome as you can, right? So the idea is, life is information. Genomics tries to extract as much of this information out of the genetic code as possible from individuals, from different systems, et cetera. And in order to do that, you really want as much sort of genetic information as possible. And you want these genomes, but you don't get genomes from sequencing. You get these little bits. And so then you have to go through this process where you take sequence reads, you assemble them into larger pieces that are called contigs, and ideally then you turn them into scaffolds, and eventually you get them to the stage of where it's a chromosome or a genome. And this is going back a little bit, a lot of these problems have been addressed over the last decade, but you still have to do this if you're ever putting a genome together. But really, it's basically when I talk to lay audiences and classes and stuff like that, I talk about, a genome assembly is a little bit like putting together the world's worst jigsaw puzzle, right? You have a jigsaw puzzle, you're trying to put it together, you don't know what the box is like, you have no idea what it's supposed to look like. So you're supposed to put this thing together, and then you're like, well, wait a minute. Like, what happens if like 80% of this is blue sky? Right? Your jigsaw puzzle happens to not have any corners or edges in it, right? Some pieces are missing. Some pieces are like just like dirty and crunchy, and you don't exactly know that it's fitting just right because the little like loop-de-loop is like bent or whatever, right? What happens when you have all these identical repeats? Um, and so you have this problem like, okay, well, how would I do this? And imagine this is not, however many, 1,000 pieces or something like that here. Imagine this is like millions of pieces. And so you now have to say, okay, I'm trying to go from a low information state, which is a bunch of sequence data, these little fragmented reads. I'm trying to make an actual genome. And how do I do that? Well, you have to you need more information. You need some kind of information that will allow you to put this puzzle together when you don't know how. And so we had this technology called proximity ligation. And so the information that I'm talking about today is this interesting kind of information that's sort of unusual and quirky. It basically tells you how far and how close every part of the genome is, and you can tell which parts of the genome are touching each other. And so if you have this puzzle, you have a bunch of pieces and you don't know how to put them together, but what if you knew that these two pieces were really close, and these two pieces were medium distance, and these two pieces were really far from each other, right? And then this piece was really close to that piece and like really far from this one over here, and this medium. And you had that for every combination. Like you had these arrows between all the pieces all over the place, right? So you suddenly have this interesting kind of like piece of information where you would say, hey, I can tell you how far away every piece of the genome is. And so I don't need the box because I am able to then basically look at this thing and use computers to tell me, put the things that are close, put them close, the things that are far, put them far. And that allows you to reconstruct that genome, reconstruct the jigsaw puzzle. And so this technology existed, but it was used for something totally different at the time. So the idea was that, so when you look at chromosomes, right, your genome or chromosomes, they oftentimes are drawn like this, but they only live this way for a small part of their life. Most of the time, your chromosomes look like this, right? It's just like a big spaghetti ball inside the nucleus of a cell. And methods were developed to try to like draw this three-dimensional structure of a genome. A lot of those tools were developed here at this department, and it's this method that was called proximity ligation. Sometimes it's called Hi-C. And what it does is you essentially take you take some kind of cell, some sort of sample. Maybe it's blood, maybe it's a plant leaf, maybe it's some bacteria, right? And you treat it with a chemical called a crosslinker. It's formaldehyde. And what it does is it goes inside and it acts basically as a molecular glue. So it's sequences that are close together within this three-dimensional hairball get glued to each other, and then they're trapped. And they're trapped next to each other. And then what you can do is you can sequence the little connections. So you have this like little red and blue, right? DNA sequence. They're now stuck together. And you can sequence them in pairs. And when you sequence one of these pairs, it tells you that whatever this red sequence is and whatever this blue sequence is, they were touching each other in three-dimensional space. Okay. And because sequences that are close touch more and sequences that are far touch less, you basically are counting how often every part of the genome is touching every other part of the genome. You're effectively getting this thing because you can be like, okay, well, I know this blue piece and this pink piece were touching each other a lot. And then this blue piece and this whatever color this is piece were not touching each other a lot. So they must be far away. Okay. So these are close, these are far. And so we kind of had this idea, and then I've been telling people, people use this technology a lot for three-dimensional genome assembly, sort of mapping the three-dimensional configuration of the genome. And I say that this technology, this was called Hi-C, was invented for the wrong reason. They were using it to look at the three-dimensional folding of a genome, which is super interesting and cool, but there's so much more you can do with this. And so periodically I'll go back and I'll say, remember, the theme is information, right? So we're catching a different kind of information. We're not catching sequence information. We're not catching, right? We're essentially saying how often do different bits of the genome touch each other. Like we're catching this interesting proximity kind of three-dimensional information of the genome. And when you have this new kind of information, you can do some really cool things with it that basically is like an elegant way to get around certain problems that couldn't be solved otherwise. So the idea is this, right? Is basically similar to what I just said. And that is that if you do this, you take cells, you cross-link them, you get these little junctions that are touching each other, and then you sequence those little touchy junctions. You essentially know how far away everything is from everything else in the genome. So if you don't have a genome assembly, if you don't have the jigsaw puzzle box, you suddenly know how far all your pieces are from all the other pieces. And so you can assemble, you can put the puzzle together based on that distance. And so what does that do? Well, what it does is, and I won't talk about this very much. I wanted to just bring this up to kind of set the stage and give a little historical context. This is what we started doing. So if you have, I showed you that reads to contigs to genome slide, right? Where you started out with short pieces, you then assemble them into longer pieces, and you're trying to recreate full chromosomes. Well, now that when you have this data set, this kind of data, this information allows you to recreate full chromosomes. And 10 years ago, this was like super hard. It was basically impossible. There were like what, like 5 genomes that were like in full chromosomes. There was like mouse, Drosophila, human, like there was like very small, like, yeast. There were like very small number of genomes that were really assembled. And it was so hard to do this. And people, there were all these technologies that tried to brute force it with, special dedicated machines that would like look at your DNA, like little spots in the DNA and stuff like that. And this was such a simple way of doing it because you can use regular sequencing to infer how far and how close everything was and reconstruct these genomes. We're working with Jay's lab on this. And I really like this approach because it was elegant. It wasn't that we had more sequencing machines than somebody else. It was this simple trick that sort of turned that field on its face. And so we started doing it for people. This is how phase got started. We started basically assembling genomes for researchers in different places. In fact, there's a fun anecdote that one day I was upstairs in the lab and a group from the Driscoll's Berry Company showed up. So Driscoll's Berries has a cool, like they love science and they have a really cool science group. And a bunch of them showed up and they said, hey, we want to assemble a bunch of our plants. But they're proprietary. So can you like teach us how to do it? Because we can't publish it. And we worked with them together. And eventually they were like, can you just like do this for us for money? We do not want to do this. And so at the time I was working together with a student who, Josh Burton, who was in Jay's lab. He was one of the original inventors of this idea that using Hi-C to assemble genomes. And so we started doing it. So actually, if you're like buying Driscoll's berries, like we did a lot of stuff for them. So like there might be those berries that you're eating, we might have sequenced them. But we started applying this for everything. And the beauty of this method was it would work on anything. It would work anything that has cells in it, like plant leaves, roots, like straight up like fish eyeballs. Like we get the craziest samples. In fact, we used to keep this like that. I mean, this is just this is a list of we started running at some point a list of papers. You can see it also ends 2021. We're now at about 300 papers with this tech. So the technology sort of took off. And people like this is now pretty standard in the assembly of genomes. So now if you're doing genome assembly, almost always you will use this technology either from us or from one of the other companies who do it, or maybe you homebrew it yourself. But this was pretty cool. So as a scientist, it's very rewarding to see something that you kind of helped birth into life, like actually take hold and people actually use it. And that was actually the reason why I spun off Face, is because I said this technology is super cool, but it's really hard. It's very complicated. Nobody would know how to do this if we just like left it to academia only, right? And so then we started doing this for folks. We started charging them absurd amounts of money for it, which was great. Then we built kits. We built all the software to do it, and so we bootstrapped Phase basically on the backs of these kind of projects. And so this is sort of example number one of like how when you get a new kind of information, you can do new stuff with it and sometimes solve really important problems in elegant ways. So, okay, so we have our jigsaw puzzle. We didn't know before what it's supposed to look like, but now we do. What about if we already have a genome? What happens if we already have a box with a picture on it? Can we use this technology to look for places where the genome has been changed or rearranged, right? Now, this happens all the time, of course, in cancer. So this is a picture of, these are colorful pictures of your quote unquote normal chromosomes. You have your two of each chromosome. And then if you look at cancer chromosomes, they are like messed up. They are crazy Frankenstein genomes, right? A lot of them have many copies, more copies than one. When they have these different colors, it means pieces from different chromosomes have swapped around. And so you get these crazy chimeric chromosomes. And this is a huge problem this is like cancer. Cancer is a disease of chromosomal chaos. In fact, we worked on this so much that we actually ended up trademarking, we own the word chaos in biotech in the US and Europe. So so we've been doing a lot of stuff around genomic chaos. And the reason why this is relevant to what I'm talking about, right? Is again, we're talking about information, and these things, again, DNA is information, and so when it switches around, when it multiplies, when it deletes, that all changes what your cell does because this is the instructions for the cell. And trying to understand now, okay, well, like this was someone's sample, right? Like this is somebody's cell. It came from a tumor that a patient had. Now, a lot of these don't do anything; they're just chaos. But some of them actually will drive the cancer. A lot of these mutations, a lot of these rearrangements, actually will drive cancer. You potentially can drug against some of them. So trying to understand what goes on in these cancer genomes is really important. And I didn't really talk about this, but this is something that happens to genomes all the time. Genomes rearrange, right? It's not only cancer. Cancer is like the most obvious case to use this, but right, you've heard of possibly of things like inversions where a piece of DNA will just flip around, right? Translocations where two pieces of DNA from different chromosomes will swap places, right? And what's interesting is this. So if you were to sequence like this translocated chromosome with regular sequencing, versus like your normal chromosomes, I don't know if they have normal ones here, or this inverted one, you wouldn't actually detect these events are happening because the sequence isn't changing, right? So if you just sequence the genome itself, you're reading the letters. This is analogous to having like two jigsaw puzzles that are the same, but in one of them, you took two pieces and you swapped their places, right? But the pieces are the same, they're still there. And so if you're just looking at the pieces themselves, nothing has changed. And so if you're sequencing a genome, usually people look at the mutations, the letter changes in these genomes, but structural stuff, when DNA is just swapping around, it's actually really hard to detect by regular sequencing. Now, remember what we had was, right? We have this quirky little piece of information that tells you what's far and what's close in the genome, right? So now, if we're looking not so much for letter changes, but rearrangements in genomes, we should be able to detect these things because we suddenly know what's far and what's close. And it's all because this assay captures the distance between different parts of the genome. And so same technology, but now let's apply it to a genome where we already know what it's supposed to be like. We have the reference human genome. Now, what happens when we do this on a tumor sample? Well, when we do it on the tumor sample, what happens is it basically it catches those structural rearrangements because you can see you can see when DNA swap places. Normally, like if you get leukemia, for example, you would go to the hospital and they would draw your blood and they would basically do a bunch of different tests on it. So this is one is called karyotyping. They look at your chromosomes under a microscope. And part of the reason why they do it is because a lot of these big, like I don't know if you can see in the slide, but like, right. So here's a piece over here that doesn't belong there. It's not supposed to be there. And so there's a chunk of chromosome that got stuck on a different chromosome, right? And you can't really tell the sequence or anything like that, but you can see that it's not there. And so for about a century, people have been using this as a diagnostic, looking at these chromosome spreads to try to detect things swapping around. And so, but again, like, you're looking at DNA with your eyes, right? Like, what are you seeing? Like, it's super low resolution, right? A lot of events, you can't see it all. So they have other assays. You have things, CMA's chromosomal microarray that can detect when sequences are being either deleted or amplified, copied over and over, right? You have this other one called FISH, which will tell you if two little bits are close together or not with these little glowy spots. But basically, you have to undergo all these different tests, and all you're trying to figure out is, are my chromosomes rearranged, right? And so what if we had an assay that just told you like where everything is in the genome? You will be able to detect all the rearrangements. And that's basically what this does, right? So we generate these little proximity libraries, except for now, instead of, a fish eyeball or a leaf, it's now, a tumor sample. And we can tell how far and how close everything is, and that allows us to reconstruct the structure of the genome and detect rearrangements. And so most of you in the room know this, but I don't know who's on Zoom. So if you've never seen one of these circle plots, they're called Circos plots. What they are basically chromosomes laid out in a circle, and like chromosome 1, chromosome 2, 3, 4, 5, etc. And these lines represent translocations and other rearrangements. So basically, here you have a bit of chromosome 1 and whatever this one is, 17, I think, rearranged, right? So you can see that. So you can see like this T-cell lymphoma sample has a bunch of rearrangements. And then this red and blue stuff, that's when DNA is being deleted or copied over. So these are called copy number variants. So either there's extra copy of this chromosome piece, or one of the copies is deleted. Now, this one is, you got a bunch of translocations. Like, this is like legit cancer, right? This one down at the bottom, this is like, if somebody were to look at this this karyotype by this assay, like, they tell me that these aren't like human karyotypes. They're like, this is like some kind of space creature, because it has so many it's so rearranged. It's crazy. And one of the things in the cancer space is, something like 95% of all cancer samples are stored in these fixed formalin blocks. They're stored in these things called FFPE blocks. So if you get a tumor sample, a solid tumor sample, they put it in this little block of, they fix it, they put it in a block of wax. And that block of wax, that FFPE sample type, of which there are like 2.5 billion samples out in the world, you actually can't do these assays on, because like, to do this, you need living cells. You can do arrays, but you can't do this. And so you can't arrest cells, and you can't really do this on a sample like this, but our assay works. And so one of the cool things about this is, again, it's not like a harder, like, there's a lot of people who are trying to like automate this. It's a little bit like, what did somebody say? That if we pursued only translational science, by now we would have optimized leech-based medicine. So it's a little bit like that, where you say, okay, we have this assay, it's not elegant, but we're just going to make it super, super fast, right? And so whereas the thing that I'm showing you, basically, you don't have to do anything crazy. You basically make one of these sequencing libraries, use normal sequencing, but now tells you how far and how close everything is. You can detect chromosomal rearrangements. So, and then why is this useful? Well, I'll give you an example. This is one of our collaborations with folks at the Hutch. So this is a study of 48 AML patients. So AML is acute myeloid leukemia. And what we're looking at is, we're looking at 48 patient samples. This is all retrospective. So this is from before. These are stored samples. What they do is they take a sample and they look at the chromosomal rearrangements by these assays that I just showed you, right? And they try to divine, hey, is this patient like at high risk of is this somebody who is needs aggressive treatment and is likely to die real quick, or is it somebody who has sort of a milder case and maybe milder treatment will work? And so they put them, they look at the sample, they characterize it, they put it into one of these categories: favorable, intermediate, adverse, right? And then if you look at the survival rate of these patients, what you'll see is that, right, the favorable folks, the intermediate folks, and this, like the red, the adverse, they're kind of dying at roughly the same rate, right? If you take the same patients, the same samples, and you use this proximity information to understand their genomic, to characterize their genomes, you can then say, okay, well, you can then and put them into these favorable, intermediate, adverse categories. Now, when we look at it, what happens is you can see this clear separation, right? This is what you want a diagnostic to do. You want a diagnostic. If a diagnostic says you're favorable, that means, like here, like none of the favorable people died at all, right? And so this is what you want. You want that segregation. And you can see that because here is the same exact samples, but this is being processed by standard of care, those like microscope-based tests. And this is using this new sequencing tech. You're able to separate them. And the reason why it's important is because, so these people who are unfavorable, they usually just get regular chemo, and the red ones usually just go straight to bone marrow transplant. And so it actually determines how you get treated, and it determines your chances of survival. And so presumably, right, like again, this is researchers only, but like if this was available as a diagnostic whenever they did this, like they probably would have, there's a couple people in this set, if you look, that probably could have been saved, or being given a different kind of treatment. So, and then the other thing is this. So, when people look at cancer samples, one of the things that's really important is when cancer genomes rearrange, they form gene fusions, which means, right, two pieces of chromosomes, like they stick together, and boom, you get like a fusion. Like the most famous one of these is called the Philadelphia chromosome, which is very important for CML. And but when you're working with these formalin thick samples, there's no way to really look at the fusions because you can't do karyotyping, you can't do those assays. And so they use assays. This is a commercial assay called FusionPlex. Here's a sample. I'm not going to give you a lot of these, but basically here's an example. This is a sample that FusionPlex, which is a commercial approved diagnostic, says no fusions were detected, and we see tons of fusions, right? Like there's like, this one is a very well established cancer fusion. There's a bunch of other stuff. You can see there's a million other translocations. And so again, it's really all about getting to this kind of like using this new kind of information to dig deeper into these genomes. In fact, there's a whole study on these. So these are 10 samples from sarcoma patients. And again, this is like FusionPlex. This is like no fusions, no fusions, no fusions. And we just see fusions all over the place. This fusion, this NTRK fusions, there's a drug for it. So if they had known that this existed, they like probably could have treated that person. So this is basically just going to point out again that, new technology, it comes in, it exposes a new kind of available information, and it lets you do things that you couldn't do before with legacy tools. And so I gave you an example of this jigsaw puzzle thing, right? So we have a jigsaw puzzle, we don't know how it goes together. Suddenly we know how to put the pieces together. We have a jigsaw puzzle, and then we have a related jigsaw puzzle that some pieces have swapped around, and now we can reconstruct that. But there's another thing we can do here. What if you have many genomes that you're looking at the same time? Okay, so I titled the talk around the microbiome technology. What happens if you have lots of genomes swimming in you at the same time? Of course, most of us think of ourselves as human, but in fact, we are primarily microbial, right? There are more microbial cells inside of us than human cells. And also in our genome, there's more legacy microbial sequence than like human genes. So we are basically just walking scaffolds for microbes. And so the microbiome, like, it's sort of a word people throw around, but it's like everything, right? There's microbiome, like in this carpet, like there's bacteria that are learning how to eat like nylon and whatever else the carpet's made out of. And so these microbes are everywhere. They're super interesting for a number of reasons. One is they relate to your health, right? Everything from kind of like your gut stuff and your gut microbiota and sort of what you eat and how you uptake nutrients. You get all these pathogens and infections. You get something like 20% of the proteins in your blood are either made or modified by microbes. And so microbes are really part of us, and of course everywhere else, right? In agriculture, in soil and water, like everywhere you go, these things are out there. And so, the study of the microbiome is often called metagenomics. So metagenomics, and like, why is it called metagenomics, and why do we need metagenomics? And the reason is that there's all these mysterious little creatures floating around all over the place, but we can't grow them in petri dishes. And so most of these microbes you can't culture, so you have to study them like in their little communities, right? You have to basically sequence everything at the same time. And these communities consist of bacteria, fungi, viruses, plasmids, all sorts of stuff. And so you basically have to simultaneously take like whatever this biome, you have to take a whole microbial population, and you have to sequence it at the same time. And it's a much harder problem than sequencing just one genome, right? So I talked about this jigsaw puzzle idea, right? They're assembling a genome is like a jigsaw puzzle. Now, what if you have not one jigsaw puzzle, but what if somebody gave you like 1,000 jigsaw puzzles and dumped all the pieces together and mixed them all up and then said, okay, assemble these 100 jigsaw puzzle, right? Now you don't even know which pieces come from which puzzle, right? In addition to not knowing how to put them together. And so, so imagine you have all these bacteria, right? The first thing you do is you extract DNA from them, right? And when the moment you've extracted DNA, you've lost some really important information that you're never going to get back, right? Again, the DNA gets shredded into little pieces. And also, if you have like little extra chromosomal elements, for example, plasmids. So plasmids are super important. They're these DNA circles that carry toxicity genes, that carry antibiotic resistance genes. They're one of the reasons why antibiotic resistance spreads like wildfire, because these little plasmids can jump from bacteria to bacteria. There are also viruses that infect bacteria called phages that I'll talk about later. But as soon as you purify DNA, all that's lost. You don't know, like this little circle here, if it's one of the sequences, you don't know where it came from. Like, how would you know that this green plasmid came from the green cell and not from the purple cell, right? Nothing tells you, because as soon as you purify that DNA, they're disconnected from the genome. You no longer know how to put them back together, right? And so you lose that physical contiguity. However, let's look at our assay again, right? So remember, what we're doing is we're taking cells, and the DNA is still inside of these little blobs, inside of these little spaghetti balls inside of cells, and you treat them with a crosslinker, which is like a glue, right? So it goes in and it basically links together sequences that are touching each other. And then you get the little touchy bits, the little junctions, and you sequence them. And so now, importantly, because you cross-link these things before the DNA is purified, so you're basically trapping that contiguity information before you lose it. So you glue everything together when the cells are still intact, then you extract. And so you're able to, so basically what that means is if two sequences are touching here, they must have been inside the same cells when the whole thing started. Okay. And so again, it's the same assay, the same idea. All we're doing is we're taking that three-dimensional proximity information, and now we're saying if they're touching, they're in the same cell. So what does that mean? So now let's look at this microbiome sample, right? Now you have these genomes, but now they're being sort of, you're getting these physical connections between sequences inside the same cell. Right? You get connections between the same pieces of the same chromosome and across two different chromosomes, but never between cells. Okay. And what that means is that you're going to get connections inside each chromosome. And also, if you have a plasmid, if you have a bacteriophage, a little extra circle, or if you have more than one chromosome, they will touch, right? It will touch the main genome. So what that means is you can now take your mix of jigsaw puzzle pieces, right? These sequences, but now you know which ones were touching each other before you mix them. So now you can be like, okay, these go over here. These belong to one puzzle. These belong to another puzzle. These belong to, and so what it does is it allows you to separate those mixed communities apart from each other. And it's essentially a sequencing data set that does this. It connects all the genome pieces together. And it also, if you have plasmids or phages, it will tell you where they came from, because like that plasmid will have been glued to the genome of the host that it was in, and it connects them together. And so So it's a crazy way to use this data type, right? Remember, originally the data type was designed to look at three-dimensional configuration of genomes, right? Like using it to deconvolve microbial communities is like a very unusual thing to do with it, but it's my favorite. And so we started doing all these things with it. And again, it's an example in how when you inject a new type of information, you could do things that are like crazy. And I'll show you towards the end, it's going to get real crazy, like in a fun way. So this was the first ever proof of concept experiment that we did back in my Treyas lab, back before Phase was even funded. We call this the soccer ball of science. I was a yeast biologist for 20 years, and so my Treyas lab is a yeast lab. So we took a dozen yeast and we mixed them together. So what we did was we mixed a bunch of yeasts together. We made like a fake microbiome, and then we sequenced them as a mix. And we sequenced them using normal sequencing and also using this proximity ligation Hi-C sequencing. And what you're looking at here is every dot, like every one of these nodes, every spot in this chart represents one of those sequence pieces, right? Contigs, or my analog to jigsaw puzzle pieces, okay? And the connections, when they're connected, but when the dots are connected, that means that those two pieces were touching each other. They were inside the same cell. Okay. And so this is what I mean by lets you separate. So if you didn't have that data, all these sequences would just be a gray mush, right? Just be a gray field. They are all disconnected from each other. You don't know which ones are coming from where. But when you have this proximity information, it takes these genomes and it puts them into these clusters. And each cluster, of course, later we look at them because we have the reference genomes. We can say, okay, well, this is one of the genomes that we put in. This is another one of the genomes. This is another one. And so this was the first demonstration that you can use this technology to deconvolve microbial communities. But for people who are kind of like more expert in this, it was also the first. We show these are eukaryotic genomes. So what that means is that every one of these genomes has more than one chromosome, right? They're all yeasts, they're all fungi. Separating apart eukaryotic genomes with multiple chromosomes is essentially impossible using normal methods, because when you have two fungal genomes and one of them has 10 chromosomes, the other one has 16 chromosomes, even if you assemble all the chromosomes completely, how do you know that these 10 go to this organism and these 16 go to that one? Like nothing tells you. And so not only was this a really cool way of deconvolve microbial communities, but the fact that you can do it to eukaryotes was really something super novel. And we had a lot of other interesting papers along that subject. Now, we can take this, and remember I talked about plasmids, I talked about phages. We can actually use this information to get a glimpse into real microbial communities that normal technology doesn't give you. So here's a figure where what you're looking at, this is a fecal sample, this is a poop sample that's been assembled, and every one of these little circles in like one of these little stars, that represents one of the genomes of some bacteria that was in the sample. Okay, so you're seeing like this is some microbe, this is a microbe, this is a microbe, this is a microbe. The blue triangles are phage. So phage are viruses that infect bacteria, right? And normally you wouldn't know where they're coming from, right? There's because again, once you purify DNA to sequence it, you get a phage, you don't know where it came from because it's not connected to the genome of the host. But here, because we glued them, we could tell that they're interacting. And then these orange ones, they're plasmids. And plasmids are really important too, because again, there are these little DNA circles. They don't have a home. They jump around between different microbes, but they're also the vectors of antibiotic resistance. So you can, and what's cool with this tech is you can see, for example, this little phage here, this little blue triangle, it's touching two different bacteria, which means you can detect when you have one phage that's infecting multiple microbes, or if you have a plasmid that's jumping between different microbes, that's called horizontal gene transfer. So normally when you do a microbiome experiment, you sequence your sample and you see something like this, where the different colors represent the abundance of different microbial groups. So you can be like, I have, this much of bacterial genus A, this much of bacterial genus B, etc. It's a little bit like looking at the earth and saying, okay, we got like 60% insect, like 50% plant, right? Like 20% bird. That's sort of like what the normal way of sequencing is. But this is what's actually happening in these communities, right? They're much more complex. It's not just like the abundance of some genus or taxa. So we can start seeing these mobile elements, this mobilome of these microbiomes, without having to do anything crazy. It's just a sequencing trick. Okay. Now, what's interesting about this study? This comes from a cool study where this is from a fecal transplant. So a fecal transplant is when somebody has a disease, like, and then you basically you like replace their poop with poop from a healthy person, and it like cures them usually, depending on the disease. So this is the donor. This is fecal sample from the donor. This is like a healthy poop. I'm going to show you the next slide is poop from a person who's on antibiotics who's about to get this transplant, and their thing looks like this, right? So this is a patient who has been sitting on antibiotics before getting a fecal transplant, and so and you may notice subtle differences, but basically this sample is dominated by a small number of gram-negative bacteria, right? Remember, antibiotics wipe everything out except for things that are resistant to the antibiotic, and so like whatever this bacteria is, it's got all these orange pieces. These orange diamonds are plasmids. They're all carrying antibiotic resistance genes. So you just have very resistant, effectively superbugs in this person's gut, right? And this is why people get colitis and things like that, because you basically the community, the microbial community is killed, and then you have these microbes that are packed full of AMR genes, antibiotic resistance genes, that dominate. But now what we're going to do is we're going to do the fecal transplant. So now we're going to take the healthy poop and we're going to put it in this person with a sick poop, and it goes back to normal, sort of, except for it's not entirely normal. And I'll show you why. So first of all, it's very interesting when you're essentially switching environments, right? You're taking all these viruses and bacteria and all this other stuff from one person and putting them in a different person. So it's like taking a bunch of invasive species, putting them on a new island, right? And so you can start seeing a lot of these viruses start jumping to different hosts because now they have different bacteria they can infect. Things start moving around. The other thing that we noticed was, you see this E. coli, this is Escherichia coli. It has lots of plasmids. So something happens during FMTs, which people didn't know about before. And that is that for whatever reason, you're always left behind with at least one gram-negative, usually E. coli, that's just full of AMR, antibiotic resistance plasmids. So, what it does is it acts as a reservoir. So the person is healed, quote unquote, physiologically. You get the fecal transplant, you feel better, you go home, but you still have a bacteria in your gut that is carrying tons of antibiotic resistance genes. It's just not hurting you. It just lives in your fecal samples, and it's just acting as a sponge for AMR genes. And then what happens is it starts giving them away sharing them with its little friends later. And so you end up getting flare-ups of antibiotic resistant microbes later. But that's this thing that we could only see if we can tell which plasmids are coming from where. So the short story is, again, new information. We're now connecting all these little mobile elements to their bacterial genomes, phages, plasmids, etc. What are we going to do with this? And this is going to be the fun part of the talk. So we started saying, okay, let's go ambitious with this. Because normally, if you're working with bacteriophage, it's very hard to isolate them because they only live inside their bacterial hosts. What if you have a bacteria that can't grow on a petri dish? How are you going to get a phage against that bacteria, right? But our technology lets us do this. And so here's an example. I wanted to show you a different sample type. This is from a cow. This is rumen from cattle. And you can see like there's a million of these little stars, right? So those are all bacteria. Almost all of them are novel. Remember, what this technology allows us to do is to take a microbial sample without any prior knowledge, reconstruct tons of genomes for bacteria that were in there, and also connect all these, right? So all these blue triangles, those are viruses. Those are phages that are infecting those bacteria. And so suddenly, from a single sample, we now have over 1,000 virus genomes, and we know who they're targeting, right? And we did this all from one sequence run without having to do anything. And I don't know why rumen's got so many phages. but that seems to be the case. Now imagine if I had thousands of samples like this. And I should also tell you that at the time when we generated this data, because I'm not a phage person by training, I said, hey, how many phage host interactions do people know about, the literature, right? Like in the field. And it was like 300. So from a single sample, we're getting more phage interaction data than the entire world has known about. And we had tons of these samples. And so we started doing them on all sorts of things. We did it on here's a paper we published with Pete Gergis's lab at Harvard. This was a sample from hydrothermal mass. When you start studying microbiome, you go kind of crazy because suddenly you're like, I want to sequence the bottom of my shoe. I want to sequence the bottom of the ocean. I want to sequence what that bird is eating. I want to sequence beer, right? And so you're there's crazy microbes and viruses everywhere, and we want to find out about them. This was a super cool study. These are samples from hydrothermal vents, those super deep ocean chimneys that spew out sulfur or whatever, and so they got little biofilms from these chimneys, and we found all sorts of new stuff. One of the cool things was for phage aficionados out there, was that there were a lot of phages that would infect not just one bacteria, but across even phyla. So they had these phages that would infect. Normally, phages are very specific. They attack a particular microbe. These babies were affecting all over the place, right? And so they did a lot of work to prove that it was real. Nature Microbiome made them fight for it. But we started doing all sorts of things, right? So remember, we're a company. We do this as a service. We sell kits. And so people come to us with projects. During all the research projects, we're aggregating all this data. So we started doing all sorts of things. We were doing cows. We were doing fungi. We were doing underwater things. We sequenced the poop of a celebrity YouTube cat, just anything we could get our hands on. And it was great. And over the years, we built the world's largest collection of phage host interaction data. So at some point, we got a bunch of funding from the Gates Foundation where I just said, listen, can you just let me sequence bazillions of samples across the world? And so we had a big grant, basically collecting wastewater samples from across the world and sequencing them, and fecal transplant data from all over the place, animals, all sorts of stuff. So we now have this massive repository of phage genomes, and uniquely, we know who they interact with, right? So we don't just have the genome of the phage. We actually know who their hosts are. And then, of course, like everybody else in today's world, we used AI to train on that data and predict even more. And so we have millions of phage host interactions, either ones we've captured ourselves or ones that we inferred from these predictive models. And so why is that useful, right? So we want to do useful things. Like when I talked about the cancer thing, I showed hey, look at that. We can find things in the person's genome that might lead to them getting cured, right? What do we do here that's useful? It's interesting. It's cool science. What do we do? So one of the things that phages are used for, because remember, phages are viruses that prey on bacteria. So there's a whole field about using them as antibiotics because a lot of bacteria are becoming resistant to normal antibiotics. This is a huge problem. And phages potentially can be, well, not potentially, we know this. They've been used for 100 years to kill bacteria as a medicine. So there's this burgeoning field of using phages to kill bacteria, right? Here's a phage, it injects its DNA, it replicates, and it bursts out of the bacteria. Boom. And this is a technical schematic of how antibiotic resistance works. But I'm going to show you. So I'm going to show you the next slide. This is one of my favorite slides. This is a 10-second video of a bunch of bacteria floating around on a microscope slide, and a particular protein is about to be added to this milieu. And let's watch what happens. So in about 10 seconds, this protein explodes like all these bacteria, right? It wipes them entirely out. And it's not a phage. It's a protein that I didn't even know about beyond basic biology. So when I showed you that picture of phages, they're always drawn this way in textbooks. Like phages, they like explode from the back, like they infect bacteria, they replicate inside the bacteria, and then they blow up and they escape. But they don't just blow. I thought they just blew up. That's what happened. But actually, no, there's a special protein that they make called a lysine. And this lysine protein, that's what the phage uses to break out. So it's lysine protein because it lyses bacteria. And so bacteria the phages make a protein called a holin, because it makes a hole. And then it makes another protein called a lysine, and the lysine lyses the bacteria. It basically pops a hole in the membrane, and the bacteria explodes. You can actually see that in that little video, right? Like when it's doing this thing, you can kind of see it's going to pop a hole and it goes bleh, right? The cytoplasm of the bacteria just bursts out because bacteria are like little balloons. They're pressurized inside. And so what's cool is this protein actually serves as a super effective species-specific antibiotic. So if you take that protein and just purify it and sprinkle it onto some bacteria, its host, it's specific to the species that it came from, it just murders it. So basically, all these phage genomes, right? Remember, every phage has to have at least one lysine because it has to somehow replicate and get out of the cells. So if you go into these phage genomes, you can find the lysine gene, you can express it, you can purify it, and then you can use it to kill its host amazingly. I want to show you something cool. So this is one that we've identified, because remember, we have millions of these things in our database. So we went through, we identified it, we screened a few, we identified one against vancomycin-resistant enterococcus, VRE. VRE is one of the most common causes of hospital-acquired infections. This protein works at like every pH, killing VRE. It's stable at 37. You can leave it in human serum for a long time. It still works. So basically, these are like great antibiotic proteins, right? And it doesn't kill everything. It just kills that bacteria that the phage came from. But here's the thing. Those of you in the audience who are familiar with microbiology, so when I say it works in every pH, what does that mean? So this blue bar, this is measuring what's called OD, optical density, how cloudy a culture is. And we are starting at OD 0.6, which is about log phase, and then it goes down to OD point nothing in about 15 minutes. So this is saying we're starting with a culture that looks like a beer, like a cloudy beer. We're going to sprinkle some of this protein in there, and in 15 minutes, it looks like apple juice, right? That's what this data is telling us. That's how potent these proteins are. And then, and the other cool thing is, it kills every resistant clinical isolate that we put it on. So these are a bunch of clinical isolates of VRE. They are resistant to vancomycin. So V stands for vancomycin resistant enterococcus. So here's an example. There's a lawn of bacteria on here. If I sprinkle vancomycin here, nothing happens. This strain is resistant to vancomycin. If you had an infection and eat a bunch of vancomycin, it would not kill this bacteria. But the lysine still kills it. That's what that little spot on the plate is. Okay. So these lysins, they don't care about antibiotic resistance, at least not against the normal antibiotic resistance. It just kills every strain of this. So they're super cool. They're species specific. And we have a bunch of other projects that we're working on, some of them completely unexpected. So for example, you can use these proteins to kill methane producing bacteria in cow guts. Remember I showed you cattle rumen? Like, why are we working on cattle rumen? One of the reasons is cattle rumen is actually one of the major sources of greenhouse emissions, right? Because methane is way more powerful than carbon dioxide. So if you reduce like cow burp methane by like 50%, that's the equivalent of taking every car off the road. Like the numbers are crazy on these. There's so many cows, there's billions of cows. They're producing like, tons and tons of methane. So if you can knock these down, it will create a real significant change. And so we're working with Gates Foundation, another foundation called Grantham. We're collaborating with a company in town called Lumen. But basically you can use endolysins to kill these methanogens super effectively. We can actually reduce methane. I mean, this is not in cows yet, but but basically, yes. So you can knock methane down. Lumen actually has this other cool thing that just came out. So this is not our work. This is all Lumen's work. They actually made an endolysin against C. diff. So C. diff is the bacteria that causes colitis, when antibiotic-resistant colitis. And so what they do is, so Lumen uses spirulina to express proteins. Spirulina are these cyanobacteria that you can eat, right? They're like little probiotics and whatnot. And so if you just make a transgenic one that expresses a lysine against C. diff, 100% cure rate. So you just eat the spirulina and it makes the lysine, and the lysine kills C. diff in your guts. And it's super effective. And there's a million other things to do here. What I really love about this project is because we now have so many of these things. And they're all like little molecular machines. They're not antibodies, but they're kind of like antibodies against bacteria, right? Because they know they specifically bind their target and they specifically pop their target. And there's a lot of information that you can glean out of them. Because they're phage proteins, they're simple, they're small, they don't have like multiple subunits and all this stuff. So it's a very interesting protein design problem, and that we're doing a lot of work on it now. And now I'm going to come back to the beginning. So remember where I started? I started talking about the technology that was originally intended to look at how a genome folds in 3 dimensions. And because it was giving us this special information that tells us what's touching in the cell, we can use it to do all these other things: build a genome, look at rearrangements in chromosomes in cancer, discover new bacteria by the thousands, right, from individual samples, even come up with like, like a completely new, like a new antibiotic paradigm. So who would have thought that using something that was originally designed for that could lead to these kind of new applications? So that's gonna be my time. Here's a picture of Faze being cute, and yeah, and I'll take your questions. And I guess there's a social thing afterwards. Yeah. Oh, we're doing Zoom, so.
First of all, thank you so much for your talk. That was super interesting. I was just wondering since it's on proximity, does it illuminate the capacity for like epigenetic markers or like alternative splicing? Because to my understanding, and I could be wrong, some epigenetic markers like methylate or basically change the density of how things are wrapped.
Yeah. Yeah, and that was the original. That was why this assay was developed originally, is to look at that 3D folding is one of the kind of epigenetic traits of the genome, right? Like certain parts, if they're tight or if they're loose, if they're bundled up, if they're not bundled up, right? You can have these zones, these domains that have like more of a particular, let's say, transcription factor or methylation. And so yeah, so there's like a two-way relationship where like the three-dimensional folding of the genome affects epigenetics, but also epigenetics, like methylation, affects the three-dimensional genome. So, right, so if you methylate DNA or you don't methylate, it changes the three-dimensional structure. So yeah, so there's that's a really good question. That's exactly what this technology was originally intended for. And so yeah, so having tons of these genomes, tons of these data sets really helps us also understand, to those who are interested, how the three-dimensional folding of the genome kind of interfaces with other facets of biology like epigenetics and stuff.
Thank you. That was really interesting. I think one of the questions that I had was about potential combination with single cell data, especially like for the gut data that you were showing. Because I can imagine, I mean, like it's really great to be able to get this detailed information just using like bulk sampling. But have you looked at combining it with single cell, or do you have any ideas about how those two could?
Yeah, so it comes up periodically. We haven't directly done it with single cell. Single cell is for microbiome, it's super powerful. Like we talked, like the JGI folks do it a lot, so we interact with them a lot. I think what the two assays give you slightly different. It's like there's like a Venn diagram of what each assay gives you, and there is some overlap and some non-overlap. Like there are certain things that like you can get with this assay better than with single cell, and there are certain things that single cell will be better at than this assay. The short answer is we haven't directly daisy chained them to each other, but they're very complementary to each other.
Cool. Yeah, that makes sense. And if I could sneak in one more, one of the things I was kind of surprised about with the unhealthy gut was that it seemed like the different populations of bacteria that you had didn't all like share the same plasmids that were leading to this antibiotic resistance. Like a lot of them had unique, which I guess maybe is a bummer because if in theory if it were just one then you could just like go after it, but at the same time maybe this makes it more valuable because it's not just like, oh, like one, it's really individual, like what's causing the
Yeah, I think so. So there's a bigger story there because so that was one. We have like there are like hundreds of patients there. We see a ton of there's a lot there it's part like I showed just those data sets because I didn't want to like I wanted to make the story a little bit more like kind of light for different audiences, but like so that E. coli thing that I showed, where E. coli after an FMT is left behind with a bunch of different AMR plasmids in it, that happens in almost every patient. That's like a phenomenon that nobody knew was happening before. We've done a lot of work looking at those data sets where we have these things where they'll give the same donor material to a bunch of different patients and they all engraft different things, which is like super cool and we don't know why. It's really interesting. When we tried looking for like what like you can say like, okay, are plasmids always staying in the same hosts? Are they jumping hosts? Are they spreading? Are they basically, we started cataloging how often the different events happen of things changing or things being unique or not unique. Basically, everything happens. There are examples of everything. A lot of them will stick around. A lot of them will completely change host. A lot of them will appear and disappear. The other thing is at this resolution, this data begs a question of what is the same, what is not the same. Like, what does it mean that it's the same plasmid or not? Remember, the vast majority of these things have never been sequenced by anyone. So no one even knows what, like the plasmid databases are terrible. So if you have two plasmids in, they're never going to be the same because these are natural communities, right? They're evolving as people grow. So at what point do you start calling them not the same, right? And so there's just like a lot of complexity within that question. And the answer is we see both. We see some things that are shared and some things that are not shared and everything else. And so the most obvious pattern is that gram-negative AMR sponge. But there's like so much complexity going on.
Going back to your first cancer karyotype, it seemed like some of those chromosomes were resistant to rearrangement, like number 5 or something was like all solid one color. Is that random or is there any rhyme or reason why that might be?
I don't know. And I imagine if there was an actual like cytogeneticist here, they might tell because there's a lot of the AML stuff. They've probably done hundreds of thousands of millions of samples. It's probably somebody knows the answer to that question. I just don't. The other thing about resistance, like it's a question of like, what do you call like? Do you say like, do you take a bunch of individuals and say, because you're basically taking a snapshot in time and you're saying, okay, none of them had a rearrangement in this gene, or do you need some sort of a time course where you're saying, is it actually resistant to it happening, right? So there's kind of like, but maybe before, so I don't go crazy with this, but like they're certainly known to be hotspots of rearrangement. A lot of times it's also the fact that it's being selected for by the tumor. So you might be getting rearrangements all over the place, but cancer only happens if you hit this thing. And so you're only ever going to see that thing. So it's not that the other ones are resistant to rearrangement necessarily. It could be that simply there's not as much sort of oncological, effect to them rearranging, and so the cancer doesn't select for them. And but I'm just not knowledgeable about that at all.
I just wanted to hear you say something about your phage-based antibiotic scheme. Is there a non-GMO way to do that, or is there some?
Well, these are natural proteins, as you know.
Yes, but they're not
Yeah.
natural in that organism that you're putting them in, right?
Well, the way you do the non-GMO is you just purify a bunch of protein and eat it.
Okay, but that works? You don't just digest it?
In some cases it works. You can also encapsulate them in little pills that
I
don't
see.
digest, right? It works like, it works for injection. It works. So there was one of the people we work with, they had a company called Contrafect that for 12 years, they basically brought all these license through phase 3 clinical trials, and then they collapsed during COVID. But basically you have like license have actually been way further down the clinical path than any phage.
And it's via some pill that only burns.
Well, I think that one, you can do it through pills, you can do it through creams, you can put them in topical things, you can put them in aerosols, you can put them in, you can, you can put them in, like COVID vaccine, mRNA, LNPs. You can, yeah, basically anything you can do with a protein, you can do with these things.
So I think you kind of answered my question, but how do you know that the license only work against one specific type of thing when you have these communities of multiple type of microbes?
Well, in the real answer to that question is you'd have to test them, right? I mean, every protein is going to be different. You don't actually know that it only works on that thing until it's been observed. So, for example, if you're and they're different between gram-positive and gram-negatives because of the cell wall structure and stuff. So like gram-positive one license in most of it in the literature, usually they are very specific to the species. Not strain specific, but species specific. Gram-negative license tend to be more broad, so you can get one that hits a bunch of different faraway species. So you don't know exactly for each individual protein until you check, but it's just historically that's what's been seen.
Okay.
So with the cancer chaos, your assay, are you trying to get it approved for like testing? And where?
It's a long process, but that's certainly the direction we're going. We're doing lots of like large studies. Yeah, I mean, I didn't go, again, this is really 3 talks squished into one. But yeah, we have all these like it's 100% concordant with standard of care. We find all this new stuff. There's all these statistical analyses of like basically it reconstructs everything the other assays reconstruct and finds the stuff that the other assays miss. And I think we're doing studies that are like 300-400 sample patient cohorts and stuff like that. So
I was really more wondering, like practically, how would it work? I mean, assuming you get it approved, would they be sending you samples, or is it something you license to?
To a particular CLIA lab. It could be, wherever that happens to be. Like we're partnering with a few CLIA labs now because setting up a CLIA lab is kind of a bear. So it's like a business question of like, how do you even start a diagnostic company? You like somewhere there needs to be a CLIA lab for someone to run it, right? Again, we do it as services, we do it as kits. We built a whole software pipeline that makes it very easy for people to do that analysis themselves. And so we're making it portable so that, like labs can use it. Actually, like the UW cytogenetics lab uses it. So it's going into the diagnostic direction. The question of whether we partner and license it out or just keep it ourselves is, sort of up in the air. We're trying both a little bit, but yeah.
Maybe this is the last question. I was not aware that most of the microbiome species are unculturable, which is really interesting. So I was just curious. You mentioned that some of the stuff you found was not like you didn't have a reference genome for it, but I was curious if you found any like mysterious species where you like don't even know how to characterize it.
Yeah,
or like
all the
felt it
time.
new.
We have species like so when I say unculturable, it's not necessarily that we just haven't figured out how to culture them yet, right? Like in the gut, I think most things have been cultured. But like you grab soil bacteria or whatever, good luck. A lot of them also are just simply they're anaerobes, so they are just hard. Super hard to culture. Those methanogens that we're killing, they need to live in pressurized hydrogen. Like it's impossible. Like finding a lab that will do those experiments is crazy. But to answer your questions, we find crazy mysterious stuff constantly, like things where it's a genome, it's a mag, it's got genes, you can tell that it's a complete genome, every database says, I have no idea what this even is. So it happens like all the time. We don't even like, yeah. It's well, so I don't know if you guys know this, there's a software package called GTDBK. It's the Genome Taxonomy Database. It's like one of the ones that people use for microbiome analysis by a lab in Australia, Phil Hugenholz. We processed a bunch of like wombat and koala poop samples for him. And he's like, with these 4 samples, I think it's like my next 3. Like he was like, there's stuff in here nobody's ever seen. And so it is really cool. It's part of what makes this technology really interesting is that it does do that, is that you're like that's why I'm like sequencing like crazy random samples from all over the place, is that you find like super novel stuff all the time.
Okay, let's thank Ivan one more time.
Thanks, guys. Thank you for coming to my talk.